An embryo is an unborn or unhatched offspring in the process of development. Embryogenesis is the process by which the embryo forms and develops. This model will focus on the development of a human embryo. Human embryogenesis lasts approximately 8 weeks after fertilization. Fertilization involves the fusion of male and female gametes in order to form a zygote. Fertilization in terrestrial animals, including mammals, is generally internal. This allows for a moist environment through which the sperm can move toward the egg. Mammalian eggs are cloaked with a special type of cell called follicle cells. A sperm must travel through this layer of follicle cells before it reaches the extracellular matrix of the egg, known as the zona pellucida. Part of the zona pellucida functions as a sperm receptor. The sperm binding to this receptor allows the sperm to pass through the zona pellucida and fuse with the egg cell, allowing the two cells to merge genetic information. The merged cell then begins a process known as cleavage, in which it divides without growing. The cell divides into 2 cells, then to 4, then 8, then 16, and finally into 32. This all happens in the zona pellucida. Cleavage stops at this ball of 32 cells, which is now called a morula. Fun fact, morullas are named after the Latin word for mulberry, due to their appearance. The cells then cluster together in a process called compaction. At this point, the cells start to become a little bit different from each other. The process of cells becoming more specialized is known as differentiation. The ball of cells then begins to secrete a fluid that forms a space in the center, resulting in a hollow sphere of cells called a blastula. This process is called blastulation. Towards the end of blastulation, the zona pellucida begins to disintegrate. The next step in the process is gastrulation. Animals are essentially built around a digestive tract, and gastrulation is the process that forms this tract. To begin, an indentation known as the primitive streak forms between the oropharyngeal membrane and the cloacal membrane. In a process known as invagination, this part of the blastula folds in on itself. The place where the blastula begins invaginating is called the blastopore. For more complex animals, such as humans, the invagination continues, creating the archenteron, which serves as the embryonic cavity, until another hole is made on the other end. Human beings are deuterostomes, meaning that the first opening, the blastopore, becomes the anus, and the second opening becomes the mouth. In contrast, the blastopore of protostomes, or simpler organisms, becomes the mouth. Cells then use the primitive streak to migrate from the outermost layer of the embryo, called the epiblast, to the interior of the embryo through a process called epithelial to mesenchymal transition. This ingression forms 3 distinct layers, known as germ layers. At this point, the embryo is now trilaminar. These different layers go on to form different tissues. The outermost layer is the ectoderm, which gives rise to the epidermis. The middle layer is the mesoderm, which gives rise to somites, forming muscle, bone, blood, and blood vessels, etc. And the innermost layer is the endoderm, which gives rise to epithelium.